Thank you very much, Huachi, to share this information with us. Please keep asking questions in the Q&A box. The speakers are available and they will answer the questions in writing. We will take some on later on. So our next speaker is Professor Jose Manuel Sanchez-Vizcaino. He is full professor at Complutense University of Madrid and director of the reference laboratory of the World Organization for Animal Health, OIE, for African swine fever. Most of us, or all of you, know him, but to talk about his scientific interest, he is currently focused on the study of epidemiology and preventive medicine of infectious diseases of animals, as well as of the development of new diagnostic techniques, new generation vaccines, and new strategies for their control. He is actually currently the coordinator of the EU VacDiva project for a safe and effective vaccine against ASF. And we would like to hear about the recent advances on ASF vaccines inside this project. Jose Manuel, the floor is yours.
Good afternoon, dear colleagues and friends. It is a real pleasure for me to be with you all today. And I would like to thank very much the organizer for these invitations to share with you what we did and during this year and a half of VacDiva already projects. So I'm going to present for you Festival VacDiva for the ones that are not very familiar with it. This is a partnership with 13 partners. Two industrial company, one is Ingenasa, the other is MSD, and all of them from the European Union. But also we have other colleagues from outside of the European Union that are member of our But also we have other colleagues from outside of the European Union that are member of our project as well, like the people from China and also the people from Kenya, the ILRI Institute, and also the Vladimir Institute in Russia. So the objective of VacDiva, probably some of you already know, but most of you probably not. I'm going to review it for you. The first objective is to provide a safe and effective vaccine for wild-boar and domestic pig ready to registrations. This is important. The second one is to develop a VacDiva test that allows us to distinguish between the vaccinated animal and the infected one. And finally, the third objective is to design an ASF control and eradication strategy of the different epidemiological scenarios that today occur in African swine fever in 4 continents. So these are the main objectives of VacDiva, and I'm going to introduce you now our 3 candidates. This is our 3 candidates of vaccines that we are working already. The first one has been isolated in Latvia in the 2070, and in one wild boar that was shot, and we call RIE1. It's a genotype 2, naturally attenuated, not hemoabsorbing, protection proved against genotype 2, and we are already working very much in the cell line adaptations. It's already working in progress with good results. And also because of the structure, we have an exclusive genome that allowed us to have VacDiva, as I'm going to show you a little later. The second candidate is an old candidate. This is an NHP68, has been isolated in Portugal, and is a genotype 1, naturally attenuated, not hemoabsorbing, protection proof against genotype 1 and genotype 2. It's already adapted to cell line. It's growing beautiful, and we are very happy with that. And also, we have VacDiva working with this virus. And the third candidate is ASF and African swine fever, a REACT CV1. Is a genotype 2. It's attenuated at the laboratory with cell culture, so it's adapted perfectly to the cell line. And we don't have results and protections in vivo because we are still evaluating the attenuation in vivo. With these three partners, we also have other selections mutants that are, some of them are already ready to be used and in vivo study, like the three ones that we have from RIA1, from this one. And also we are very good progressing in have a mutant for the NHP68. So the goal, as you can see, is to obtain a safer vaccine with VacDiva properties. So I'm going to summarize for you because we don't have too much time. The objective, according with the objective, the results that we have already done. So we're going to start with the fair objectives, that is the in vivo studies and domestic and in wild boar. Also, we're going to tell you some vaccine manufacturing research that we already done. The first one is the studies that we did in domestic animals with the Lavia 17. So we has been evaluated 3 different immunization routes and 2 different doses per route. So here you can see we have 3 routes for immunizations with different doses. So after the challenge with we challenged with Armenia 07 with 10 HAD 50 by intramuscular route. After this challenge, the best result of all these 3 studies has been performed with the route number 2 and 100 CCDI doses. So this is in relation with Lavia. I'm going to show you this result a little more clear to show you why the route 2 is the best route for immunization, as well as the doses of 100 TCDI. And here, in this average of clinical score, you can see perfectly the number 2 is the one that have much better results with very, very little or none clinical score or problem. So you can see here the survivor was very, very clear with very few little viremia, and the control, the naive control, was absolutely with a very high pathology. So the summary of this study is to have a very strong seroconversions of 100% protections against Armenia 07, virulent Armenia 07, with very slight or no clinical signs at all, negative or very weak intermittent viremia, as I showed you before, occasional virulent virus shedding in some animals, 2 of 14, even absolutely an absence of clinical signs. The controlled naive pigs die within 10 days with very severe acute form of ASF. We also have already 3 the latest mutants ready to be starting in vivo assays. So these are the 3 DA, DB, DC, also from Lavia. So we're going to do next week, we're going to start with this experiment in vivo. So we also performed in domestic a comparative study of the NHP68 in the origins way and the NHP68 adapted to tissue culture. So we compare the study in 2 groups of animals, 5 animals for the original NHP68, another 5 for the adapted to tissue culture, with the same doses of infection and the same route of immunizations. And they were being challenged with Armenia with the same doses, HAD 50 by intramuscular route. And so also we have 3 naive animals as a control. So the results was very interesting because after vaccination of both groups, we found that clinical signs quite different. We make a very significant minor side effect in the one adapted to the tissue culture than the original one, with a very good antibody response in all the animals and both type of animals, and very weak or intermittent viremia. After challenge, all pig survivors and showing transmissions, slight mild fever in most of the pigs, weak intermittent viremia with a very high CT, 36.64, and occasional virus shedding in some of the pigs in both groups. The control, as I mentioned before, was within 10 days, they die with severe acute ASF form. So here, I summarize for you what I explained you here. So you have the original NHP68 and the one adapted to cells. So you can see that, the one adapted to cell here and the original one down. So in the first one, you can see that the clinical finding associated was very, very, almost nothing, and 4 of them nothing. 1 of them very slight chronic little lesions. And in the other one, in not adapted to the tissue culture, we have worse results. In the post-challenge, again, we have a much better results in the adapted to the cell culture than to the original one, and both of them with 100% protection. So we are working a little more in that to finish with this little clinical finding associated. So in the last candidate, the results that we have so far is only in vaccinated studies. So we are not making challenge yet. So only with the vaccine of the 10 animal and 10 pigs inoculated intramuscular with the vaccine, with this vaccine in vivo ASF ASFV with the 50 passages already, we still have mortality in 1 of the animals. So from 10 pigs inoculated with this vaccine and 4 contact pigs, and 1 of the inoculated with the vaccine died of ASF. So we still have mortality with this prototype. The mortality rate is at 10%, and we are working more and working more in the attenuations to have perfect attenuations and later make the challenge to see the level of protection. I'm going to move now to the other experiments that we perform in vivo, and these occasions with wild boar. So in wild boar, as you know, as you probably remember, we published already one of the first oral vaccination of Eurasian wild boar against African swine fever genotype 2 with the Lavia 17 and by orally. And so, and these experiments, we found and described the attenuations using 9 animals, the immunizations with 5, and also the protection with other 4. So in the attenuations one, you can see that clinical score was very, very low. The one only with fever. The fever never reached higher than 40.5. As you know, the temperature in these animals is 39 to 40. So this really was very, very little increase of temperature. 7 of 9 animals and 1 of 3 of the contact, and with a very, very low viremia, 6 of 9 and 4 of 5. So these are in blue. You have the vaccinated animal, and the contact are here a little more dark, and the contact have the vaccinated one, and the contact have, you see the vaccinated have a CT of 34, and the animals, the challenge animal, the control ones have a CT of 21. So as you can see, even the contact has been quite well vaccinated and protected. So here you have the level of immunization in antibodies by both ELISA and also EBT. So you can see the animal, in dark and blue, you have the vaccinated, and dark the contact one. So you can see we immunize 8 of 9 vaccinations and 5 of 5 in contact. So the protections against Armenia 07, we have in the vaccinated 8, totally immune of 9, and in contact 5 of 5, and the control, they died of 4 of them. So we have a survivor in the vaccine of 92%. So one important things that I would like to show you is the clear difference between the control animals and the immunized and contact. And so you can see that the difference is unbelievable in the clinical score, very, very big difference in temperature, as well as in the level of viremia. We also already finished the studies to find the optimal doses with the RIE for wild boar, as well as some cross protection. So the wonderful doses that we have, we are very happy with the result already, is 10-3 and revaccinations, but also we have prepared already another experiments with 10-2, because we also believe that with 10-2, it's going to be a good protections and also a very strong cross protection and vaccinated and revaccinated. So 10-3 10-2 is going to be our next target. So we're going to tell you probably in the near future. It's going to be several papers of VacDiva in the next month. You're going to read most of them. And well, in cross protection, as I told you, we challenged with Armenia, absolutely perfect. And in relations with the other vaccinations, we was very worried because, as you know, in wild boar, we don't know exactly how many baits they're going to take. We have already a baits that they like it very much and they eat it perfectly, but we don't know how many they're going to have. So we want to be sure that there is no trouble in case that one of these wild boar take another vaccination. So we did this study with animals in experimental conditions, giving more that 1 or 2 or 3 or 5, even more of that, and we don't find any other vaccination trouble. Also, as I mentioned before, we are already very, very ready to prove the new NHP68 adapted to tissue culture by oral administrations, also in wild boar. We're going to start in 1 month from now, this experiment. So we're going to have results with this new adapted and tissue culture very soon in wild boar. We also work during this period of the research project that you know is a year and a half. We also working in the studies of the base selection and designed some little differences. At the same time, I'm going to show you some of the new baits that we designed, but also tell you that the studies has been performed in wild boar farms and hunting estates here in Spain. All this experiment is already ongoing, so I can show you too much results. And in Hungary is going to be very soon started. So the design of the baits that we did was some little changes. We changed the paraffin for one more bee wax, one more natural bee wax, and the results that we obtain is that in piglets they like more the wax bait, but not too much. You see, the difference are really not too big. And in the juvenile wild boar, we find a very clear difference. They prefer the paraffin bait. So we're going to use that paraffin bait in the future. From the vaccine manufacturing point of view, we have several data that we also would like to share with you. First of all, we don't see the pig black cell are any options in our VacDiva vaccine. So as I mentioned already, we have identified few candidates that are adapted to cell line. So we're going to continue with that to have all our vaccine produced the different prototype and tissue culture and not in blood cells. So we also are working in different procedure production procedure and different BSL level 2 or BSL level 3 to see if it could be possible to produce ASF vaccine in BSL level 2. So we are developing this production procedure in both systems. And finally, also with the vaccine manufacturing, we are testing the freeze-dry or the ready-to-use. So as you know, the freeze-dry usually is more stable. And so in VacDiva, at this moment, we are evaluating our candidates in both ways to see which one will be the best. The second objective of VacDiva is to obtain a VacDiva test. And as I mentioned before, there are several candidates because the genomes change enough to have this properly. And in particular, I'm going to present you the first one that we have. It's already for RIE. It's going to be a VacDiva test. And we already identify two proteins, one in bacteria, another in mammalian cells, that both of them can be perfectly working as a VacDiva difference between vaccinated and infected animals in the future. We already finished this study with domestic animals with the RIE vaccine and with the RIE infection. With the RIE vaccine and with the Armenia infections, and they work perfectly as a VacDiva test. The third objective, as you probably remember, is the ASF vaccine and different scenarios and different epidemiological scenarios. So as you know, African swine fever at this moment is located in different places in the 4 continents with different epidemiological scenarios. So we would like it to really see how we can use this vaccine in the best way. So far, we evaluating the characterization of the different scenarios. We are progressing very, very much in the European scenarios. We are already starting with the Asian one and the African ones. And we also are evaluated the optimization of the vaccination strategy. So we are doing mathematical modeling, and the African ones. And we also are evaluated the optimization of the vaccination strategy. So we are doing mathematical modeling for evaluate very well the transmissions in wild-boar populations, as well as what are the minimum vaccinations that could cover the population with a high immunity. So we are working with this model, this second one, not only for wild-boar, but also for domestic one. So we are doing a spatio-temporal cluster analysis, also analysis of the factor that influence the cluster formations, like environmental variability or variable, habitat of wild boar, anthropogenic factors, wild animals and domestic pigs. And we are also evaluating the estimation of the transmissions by a mathematical model. So we also did during this year and a half the first online survey. We did a survey for domestic, so the vaccination scenario for domestic and also for wild boar. So we are very lucky, and there was more than 80 experts from domestic production animal, pig, and 75 for wild boar experts from different sorts, from the state's veterinarian, for the swines and wild boar vets, for the farmers, from the environmentalists and hunter. So we have been evaluating farm biosecurity, hunting biosecurity, and many other parameters that are very, very important to really analyze the potential use and the different epidemiological scenarios where African swine fever is already present. And I would like to take this opportunity to invite all of you, if you would like to participate in the second round of this questionnaire. And so just please, if you want, join and send us your name and area of expertise, domestic or wild boar or both, to email us to info@vacdiva.eu. And we're going to answer you, and we'll be very happy if you join us. So I'm sorry if I was too fast, but as you know, I want to tell you too many things only in 20 minutes. So I really would like to thank you very much.
Thank you very much, Jose Manuel. We have now heard about the experience from the US, from China, from the EU, and now we are going to the African experience.